VPROMPT is the Vanderbilt-Pittsburgh resource for organotypic models for predictive toxicology. The projects that are included are 4 different organ-on-a-chip devices. One for mammary gland, one for early limb development, one for fetal membrane, one for liver, and then finally there's a systems engineering and analysis project that ties all of those together. So an organ-on-a-chip takes multiple cell types, grows them in the right 3D arrangement and with low overhead fluid volume so they can chemically communicate to better mimic the in vivo environment of particular organs or developing organ systems. And the hope is that that is then better predictive of what would happen for actual human exposures. There's important impacts of chemicals on early development. Mammary gland is a tissue that undergoes multiple phases of development, some in utero and then during puberty, and both of those windows of exposure are important. Limb malformations are common. Fetal membrane, when it's impacted by chemical exposure, has been shown to increase the probabilities of preterm birth. And then of course it's important to include liver, where xenobiotic chemicals, chemicals that are not natural in biology, are metabolized. So it's clearly important to connect the organs because the chemicals to which cells are exposed in vivo usually pass through the liver first, and they're metabolized there. The organs on chips are simple enough, we know all the parts, we know how it was put together, that we can go back in and dissect exactly what the adverse outcome pathway was.
What we've developed is a system that will predict the lowest, safest exposure limits for a toxicant or potential chemical of interest and whether it's going to affect mammary gland. It has a very different kind of developmental pattern where there's a rudimentary organ at the end of fetal development upon birth, and then most of the development of the mammary organ actually happens during puberty. So you can see where with exposure to a toxicant, it may have very unique effects depending upon if it's on the fetus, if it's in a young teen, or even in an adult. Exposure can modulate the mammary gland.
There's been a huge amount of effort to try to reduce the incidence of preterm birth over the last 10 to 20 years, and very little progress has really been made. So our group had data that suggested that an exposure to a toxicant in an animal model unexpectedly caused the animals to have a higher risk of infertility and preterm birth if the animals were subsequently exposed to an infectious agent. And so I think by using the devices to understand what we would call a second hit hypothesis has been really among the most fruitful things that we've been able to look at. And so we're very hopeful that that understanding would provide information to the physicians that are taking care of women at risk for preterm birth using a perspective that they haven't necessarily had in the past.
There's tens of thousands of chemicals in widespread use. We need to screen through all of those eventually, and to do that we have to have automated systems that allow dosing schedules and perfusion and moving chemicals from liver to downstream organ all automatically. What's the vision for 5-10 years from now for how do we reassure the public when a new chemical is introduced and people are going to be exposed to it, that there's a reasonable expectation that that exposure level is safe. So the role organs on a chip are going to play in this, what I see, is that there's going to be a whole suite of these organ on a chip. Some for mimicking what happens for in utero development, some for adult organs in homeostasis, and then these all need to be tied together, because it's important how the liver and the placenta and the thyroid gland and the reproductive organs all talk to one another. Now, is there going to be a one-to-one predictivity that anything that affects a particular organ on a chip is guaranteed to affect that organ in a human being? No, I don't think we'd ever get there. It's going to be an approximation. There's going to be a correlation, though. And so the organs on a chip serve as a means of prioritizing the chemicals. It's about being efficient in screening those 10,000 chemicals, and this is a good way to do the prioritization. Environmental toxicology is all about prevention. No one purposely exposes themselves to the chemicals that are in our environment. And that's where organs on a chip come in. That you can take this system that uses human cells that mimics the in vivo environment of a particular organ And it's that suite that has to be developed. That's why it's important to have multiple centers testing different kinds of organ-on-a-chip devices, talking to one another so that we can figure out how to connect them and put together the package of testing that would reassure the public that the introduction of a new chemical is not going to cause adverse effects.